Hi, hello, welcome to your B News, your channel for biology, ecology, and evolution. Today I'm really glad to receive here Antonia Meyer. Hello, Antonia, how are you?
Hi Sam, I'm fine. You?
Very good. Thank you for accepting this invitation. Antonia, she's a biologist. She recently finished her PhD in the Department of Animal Ecology and Tropical Biology in the University of Würzburg. She performed a very challenging project during her PhD. She went to Africa. Actually, she went to Kilimanjaro, where she studied bees and wasps. And her study is very important, not just to know more about species distribution, but also to understand how environmental effects affected life of insects as a whole. Antonia, she's very much passionate by bike, and her favorite work is actually being in the field, looking to the bees, studying them in loco. She is also called by her Swahili friends Mama Nyuki, which means mother of bees in Tanzania. Welcome, Antonia.
Thank you very much, Sam.
Anthony, a fundamental question in ecology relies on the understanding of species distribution. In this context, it's very common scientists try to understand the factors which can affect distribution of plants, animals as a whole. Additionally, aspects related to diversity such as species richness, diversity, and ecology interaction are among the top questions in ecology. However, top-down interactions, which are a factor to play an important role on the course of species, seems to be a little bit rare on studies involve elevation gradients, and you come to fill this gap. What is known for some species, especially endothermic, that amount of energy, such as food, affects species distribution? Does it pattern remain for ectothermical animals, such as for the insects?
Yeah, for ectothermic insects, there is a very broad meaning that temperature is more important than resources because they cannot heat them up themselves, so they cannot regulate their body temperature, and therefore also temperature limits the access to resources. So in these cases, food availability is often not equal to food intake.
Okay, so what is known about the role of climate, such as temperature and species distribution? And in addition, what would be the factor limiting the occurrence of species on extreme temperature?
So first, of course, there are physiological constraints. So if it's, for instance, too cold or too hot, they cannot tolerate these conditions in the field. Then, of course, it's an indirect effect when they cannot exploit the resources because, for instance, insects cannot fly when it's too cold. So even if there are a lot of flowers, the bees cannot go and collect nectar and pollen. And then there are also effects like there are more biotic interactions when it's warm, so with parasitoids, for instance. And of course, also evolutionary processes increase when temperatures are warmer. So there are higher diversification rates.
Right. I think one important component was for your study was related to top-down control of antagonists. Can you explain that for us, please?
So top-down control can be, for instance, controlled by predators, parasites, but also bacteria, for instance. And people think that top-down control enhances species richness because, for instance, dominant species are suppressed, so there is more space for a higher number of species to occur at the same time.
So, although the previous questions may explain better species distribution at the evolutionary time, as you said already, human land use may help to explain the current species distribution. Are there reasons to believe on that? Would you expect that this occurs also in mountain ecosystems?
So, of course, land use is an important factor in a lot of studies. And of course, this is also true for mountain ecosystems if they're very impacted by land use. Yes. So I would say that it has a lot of factors. from bottom up and also control top-down control. If predators are killed by insecticides, or if you fertilize, then you have more nutrients, and so land use is an important factor. Yes.
All right, thanks. Organisms such as animals and plants, they interact with each other. A bee, for example, it's the one of the species you want to answer questions in your study, can visit flowers for food. They can collect nectar, pollen, and this action can lead to pollination. And the pollination may lead to a production of fruit. This fruit can be either later consumed by a bird or for a fish, and maybe this fish can be later consumed or finished at the mouth of a crocodile. And this is called trophic niche. Trophic levels occur at different scales. Can you explain how these occur in your study system with cavity nesting insects?
Yes, so the first trophic levels are always the primary producers, right? So in our case, the plants, and the plants serve as food for the bees. The bees go and collect pollen and nectar. So the bees are already like the second trophic level. And yeah, what you can see here on this figure. And also the second trophic level would be, for instance, caterpillars, herbivores, other herbivores. Then the third trophic level in our system are the antagonists of the bees, which feed on the bees in blue, but also other groups like caterpillar hunting wasps, which feed on herbivores, so which would be primary consumer. And also other predators like spiders who feed on other herbivores, for instance. And then the next trophic level, in this case the fourth trophic level, are for the bees the hyperparasitoids, which parasitize the parasitoids of the bees. And for the wasps, for the caterpillar hunting wasps, this would be the antagonists of them. And for the spiders would be the predators of the spiders. And then if we go one level up, the fifth trophic level, there would be again the hyperparasitoids of the parasitoids of the caterpillar hunting wasps in the middle, and the antagonists of the spider hunting wasps. And then there is even one level above. Here the sixth trophic levels would be again the hyperparasitoids of the antagonists of the spider hunting wasps. So with this system, we have 6 trophic levels, which is enormous. Yeah.
Putting everything together in your study, you aim to understand how the biotic, like resources, as you explain here to us, and abiotic factors play a role in species distribution in elevation gradient. Some of the questions you aim to answer, I'm telling to everyone here, was, do elevation rate affect species richness? Does parasitism rate enhancement differs among different trophic levels? Do the number of antagonist species in relation to the number of host species correlate with elevation? Does land use affect all trophic levels with the same intensity? And does resource availability play a role on species richness? So this was one or some of the questions that you want to answer in your study. And to continue, can you explain a little bit for us, where did you develop where did you went to answer these questions?
Yeah, so I went to Africa, and specific to East Africa, what you can see here on the map, and we went to Mount Kilimanjaro. Mount Kilimanjaro is the highest freestanding mountain on Earth because it reaches up from the savanna to, highest elevations are 5,895 meters. Yeah.
So what you can see here, Antonia, is like, is there a forest surrounding the mountain? How is there? Like, and in addition to that, how the temperature changes from top to bottom of the mountain?
So you see that the mountain is surrounded by brown areas, which are the savannas, and then there is a tropical mountain forest, So there's not really lowland forest because there are the savannas in East Africa, but then you have a lot of different tropical rainforests, and then on the top you have different alpine vegetations. And the temperature decreases strongly. So there is a temperature difference of nearly 21 degrees.
Wow. Well, that's really a lot. And how did you perform the sampling of your study?
So we use these trap nests as a model system, which are basically tubes where we put a lot of different reed internodes inside. And yeah, then we place them on different study sites for 15 months, hatch them in the field, what you can see here in the buckets, and controlled them monthly. So we checked if they emerged and controlled for new nests. And then later on, when they emerged, we collected them, cut them out and open, and identified the species, and also other parameters like parasitism rate, for instance.
You mentioned before, like how high is this the Kilimanjaro mountain? Do bees occurs in the all range, or is it limited to a certain range?
No, bees really occur from the savannas up to the alpine vegetation. But of course, above that, where is only stones and ice, there is nothing. So there are also no bees. But we really found bees up to 4,550 meters.
Here in this picture, you look, especially here in this picture in the middle, you see a vegetation in the background. Would this one be the savanna? And if yes, at which high are you here?
So this is not savanna. These are home gardens by the local chaga, and we are here standing on a grassland. So they have really extensively used grassland, super species rich. And oh God, I don't remember exactly what elevation it was. I would guess like 1,000 meters.
Okay. Wow, it's already super high. I think it was not easy to get to these places. So one important question of your study was understand how the climate affect the species distribution or the interaction between the parasitoids with the hosts. How did you collect the information regarding the temperature? What did you use? And in addition to that, you also have used a kind of index to measure the effects of land use. Can you summarize to us how you did that?
So we recorded the Kili study. Kili project was a study with a lot of different scientists. So we have very good temperature, also humidity data, for instance, and we used thermologger to record the temperatures during the whole time. And for this study, then I only used mean annual temperature. And the land use, we classified it as a land use index, and the land use index consists of 4 different parts. The first is the annual removal of plant biomass. Then we measured agricultural inputs, monitored the vegetation structure, and also the percentage of agricultural land in the surrounding.
All right.
So a low land use index is 0. This is the lowest, and the highest would be 1.
Okay, so you have been there sampling for more than a year. I think it was around 15 months, right? So you have the...
Yes.
So you have the chance to get like different seasons. How many nests and insects you captured during the period in the field? Do you remember that?
So first we did this monitoring for 26 months.
Oh, okay.
Because we had a pre-experiment where we really, maybe this is something important because I said that bees occur to 4,550 meters.
Right.
But these cavity nesting bees, which we collect with the trap nests, they only occurred up to 1,800 more or less. So where the forest zone starts. So just not to mix these things up.
Okay.
Yeah. So that's why we did then the second experiment, what we see here in the study, and this was only 15 months.
Okay. And how many bees, do you remember how many bees did you manage with? I think this method is really nice. And you can collect a lot of species and high abundance. So I'm just curious to how much you sample.
So in this period, in this 15 months, we collected 4,015 nests, more or less, so which is like 15,000 brood cells of bees and wasps. And we had 81 morphospecies of hosts, which is bees and wasps, and 49 morphospecies of antagonists. So really high diversity.
Yeah. So, and another important thing of your study was related to if elevation rate affect species richness. Huh? Can you say about these results here in this graph, please?
So the elevation was the strongest driver of species richness. What you see that in all groups, species richness is declining with elevation. But you see that with increasing trophic level, especially for the antagonist groups, you find a hump-shaped pattern, which means that where you see this hump for the antagonists on this of the spider hunting predators, there was the forested area, so more to the in the direction of the rainforest. So the bees, they like the open, warm environment, but the wasps, they also prefer the forest, so there are more species.
Mm-hmm. And do the number of antagonist species, or better, do the parasitism rate and hence differ among different trophic levels?
Yeah, so this you can see here in this graph below, and then you see that it nearly doubles, which I didn't expect. So the parasitism rate of the bees was 12%, and then in the caterpillar hunting wasps it was 24, and the spider hunting predators 45. So it nearly doubles, and for the spider hunting predators it's nearly the half. So yeah, this is really interesting that it increases, even though the number of nests goes down for the other trophic levels.
It's really, and how this relates to the elevation? How the parasitism rate is correlate to elevation? Here you can see in this figure 3D.
Yeah, you see that the parasitism rate decreases with elevation. So less nests are parasitized at higher elevations than at lower elevations. And it decreases stronger for the higher trophic levels. So you see that this decrease is steeper for the spider hunting predators, for instance, than for the bees.
And what about the number of antagonist species in relation to the number of host species here in this figure 3C? It seems there is the same pattern.
Yeah, so here you see, so first maybe the ratio means that the number of antagonists to the hosts, if it would be 1, then there would be the same number. And here we see that this ratio declines, which means that we have relatively more antagonists in lower elevations than at higher elevations in relation to the hosts. So the antagonists are declining stronger than their hosts. And this also points that the trophic interactions are more intense in lower elevations, so with warm climate.
Right. You also have tested different parameters that could be or could have play a role in your system, and you have presented here in a table in your study how these parameters affected what you found. Can you explain this for us, please?
Yeah, so here we first thing you see that the impact of temperature on species richness is important for most of the groups. And here you see also that land use was not important as a driver for species richness. And then if we go to the right, you see that resources are divided in resource abundance and resource riches. but the resources were only important for the antagonist groups. And the importance of resources increased even with trophic level. So for the antagonists of the spider hunting predators, for instance.
That's really interesting. Antonia, it seems that the pattern that you found for species richness decrease in relation to elevation does not apply to all functional groups, no?
Yeah.
Which could be the reasons why you found caterpillar hunter predators and spider hunter predators and their antagonists present a different response at middle elevation.
Mm-hmm. Yeah, yeah, that's what I already said, that we have more wasp species in the forest, and the bees prefer the warm and open habitats. And also a multi-taxon approach from the Kilimanjaro from Peters et al. Showed that, for instance, parasitoid wasps, that they had a hump-shaped pattern, what we also found for the antagonists of the spider hunting predators.
All right. And you had sampled an amazing amount of trap nesting host species and their antagonists. It seems you have made a great effort. Is there an evolutionary reason that can explain such high diversity in this study site?
Yeah, so the first is, of course, we are in tropical mountains, which are hotspots for biodiversity. Then we have different habitat types, so completely different species as well. So we have savannas, we have maize fields, then we have grasslands, this agroforestry systems of the chagas and coffee plantations. So it's not the same system. And we have broad climatic differences. And for the bees, I think that reason is also because they are close to their origin. So, which was the xeric interior of Gondwana land, and today this is Africa and South America. So they had a very, very long evolutionary time to diverge.
Mm. Is it just an aside question that came to me now? What is known about the diversity of bee species in this area or in the entire continent? Is your study one of the pioneer studies?
So there are studies, of course, so especially South Africa, but in general for Africa, there's very, very, very few things. So was quite hard, and that's why we also couldn't identify all species. So a lot of are named like morphospecies, because there is few literature about that. And for trap nests, it's the first study. So there was 2 in South Africa, but the rest of Africa, there are no trap nest studies as far as I know. Yeah.
Yeah, congrats for you and your team. It's really nice. What I think was really remarkable, in my opinion, relate to the fact that the resources like flowers did not affect the number of pollinator species, but resources become increasingly important for antagonists. Did you expect that?
Yeah, so it was one of our hypotheses that higher trophic levels are more limited by resources than lower trophic levels because of cascading effects. So they depend on more levels. And if you have a habitat with low number of resources, then this can only sustain low number of herbivores and then in turn low number of predator species. So that's why we expected it. And on top, you have spatially and temporarily limited food resources. So there's a strong selection of the pressure for the hosts to use this resource efficiently. So that's why we thought that they are more dependent than lower trophic levels. But I also expected to see this pattern in the host groups, what we didn't see. So I also thought that this increases, like for spider hunting wasps, for instance. And one reason here might be that the quality of the resources which we recorded is not as good as for the antagonists, where we could really see for the number of nests and the species of host species, where it's really nicely recorded.
It's an amazing story. Tony, I would like to thank you for being available to participate in this chat. Just before we finish, I will have a last question. We are currently observing climate change. This is fact, and there are evidence that this environmental change can affect or impact life as a whole. You mentioned in your study that climate change based on landscape perturbation, especially concerning the loss of savanna, can affect community of trap nest insects. Are there evidence that this occurs in these study sites or in general?
Yeah, so that's what we didn't find so far. As I said, we didn't see effects of land use, but I think a reason is that, and this is something I have to mention, that this area has a very nice mosaic-like structure. So everything is very close. And therefore, I think there's not yet impact, but we see that the natural savannas are decreasing and also coffee plantations expanding, for instance. So I think that there will be effects in the future. And colleague of mine, she investigated different coffee managements, and there also the ecosystem services were not affected yet, but she could already see changes in the pollinator community, for instance, that on higher intensified coffee plantations, there were more honeybees in relation to wild bees, butterflies, and surfates. So this might be that pollination services might be not as stable as they are now in the future if land use increases. And since we have the highest diversity of species in the savanna, and this decreases now, then we will lose a lot of species. And with climate change, we also expect species to climb up mountains, but the savanna is already the lowest habitat, so there cannot be species who come after them. So yeah.
You already mentioned that there is not so many studies, but we might have examples from other places in the world. Is there a way to conserve this ecosystem and still have an environment where the community, like the locals, can work in their agrosystem without impact to nature?
Yeah, so what the locals did for centuries is sustainable. So if they would go go on like this, it would be perfect, because we didn't see effects yet. So I didn't find effects yet. But of course, yeah, if there will be investors from other parts of the world, and if they will destroy these super sustainable agroforestry systems, then it's also not as diverse as it is. And if the savannas are destroyed, then there's no plan B.
It's really interesting because there are many studies now made in Europe or other parts in the US or other countries where they are going back to more traditional systems to show that this is actually good for the pollinators. And here we had the chance to show the in loco that using this traditional method is still really good. It's really nice. Before we finish, I want just to share here a bit. Maybe you want to say, I think you have an amazing time while you were having this or conducting this PhD in Africa. So how was that? Like just the overall feelings about working in such environment?
Yeah, it's super great because it's super diverse. So it's never boring. You're always in different habitats, and you can climb up the mountain in 1 week. So one day you are in the savanna, next day you're in the rainforest, and then you're also already up the mountain in alpine habitats. And of course, I also had very close contact to the people, to my field assistants, to kids who were always interested to see what I was doing. And yeah, this made it an amazing experience. So I would never ever like to miss that.
That's great. I mean, one of the aims that we have, like interviewing specialists, is really democratize a bit of the science, let the other know what is going on. And it seems that you have the chance to do it while you're doing your work. That's nice, super cool. Just to finish, we have the chance to chat a little bit in the past regarding the closure of the trap nests for bees. And here you send me a picture and I don't want to finish this chat before you explain a little bit what it here in this picture in the left side that I'm showing with the mouse.
Yeah, so the nice thing is that even though this trap nesting species are only a small part of the whole community, you already see a lot of differences. So for instance, some bees can close their nest with leaves, other close their nest with mud, others with raisins. So here you see the whole diversity of different closures, but not only from bees, but also from wasps.
Here in this figure, if the same species of either bee or wasp, they use different materials to close their nests, or what we saw here are actually different species.
Yeah, of course, every species different methodology to close their nests, and I don't know that explain that, for instance, bees are closing the nest with resins, with clay, with leaves, with silk, with stones. So they're completely different ways. Even though it's just a small part of the community, they are super diverse. And yeah, so it's also interesting just to look at this nesting behaviors, for instance.
That's really cool. And this is in the middle is how the bee look like inside the trap nest, right?
Yeah, so this one cut open trap. Yeah.
That's awesome. Antonia, I would like to now finally to thank you for participating in this chatting. And I would like also to let this channel free for you. Whenever you have news or different results from your amazing time you have in Africa, please give me a call or write me an email or just come to my office and let me know. It would be very nice to hear more from you regarding this amazing system. Thank you so much.
Thank you too.
All right. Goodbye.
Goodbye.
And you, thank you so much for watching. And we are looking forward to meet you again in one more B News. Keep connected with us. Bye.